I was a graduate student with Doug, and we had been working on the respiratory complexes. This was when membrane protein crystal structures were few and far between, and the field was in its infancy. The structure that we originally reported of this quinol fumarate reductase was, I think, the 11th membrane protein structure. And this particular paper was looking to identify how these redox carrier molecules called quinones interacted with the protein, because there was controversy over the quinone binding site. And this was one of the very first papers where crystallography started to be used as a follow-up technique to answer biochemical questions on membrane proteins. It had been just done a little bit with the reaction center. And then, as the field's progressed, it's become more and more common to do that with membrane proteins.
Yeah, well, my group was interested in electron transfer proteins and membrane proteins. We were really just starting to get into membrane protein work. And so fumarate reductase really combined these two. And this was also at a time where it was pretty challenging to overexpress membrane proteins. And so respiratory proteins like fumarate reductase were really important because they naturally occur in large abundance. In this case, it's a terminal electron acceptor in E. coli. And so we started this collaboration with Gary Cecchini. And the first big advance was Tina's structure of the protein fumarate reductase, which was a tremendous accomplishment, both in terms of the scientific impact, but also the technical degree of difficulty. And so, as Tina was mentioning, as sort of one of the questions that came from her work was the nature of the quinone binding sites. And so this particular paper really addressed that in a very clear way by looking at the binding of these different inhibitors and characterizing them at high resolution. So yeah, so it was a really pretty cool, both the initial work and then this follow-up.
Well, one of the controversies, I guess, in the field were, I'd mentioned there are these small molecule quinones, and they carry electrons between the respiratory complexes. And it had been questioned, it appeared that there could be two binding sites for these quinones, and it had been questioned if they were both physiologically relevant. So looking at these inhibitors, they all seemed to bind to just one of the binding sites, which suggested that even if quinones could bind to both binding sites, that this one binding site was likely the site of electron transfer.
Quinones are just fascinating molecules. As Tina was mentioning, they carry both electrons and protons, and they're membrane soluble. And so these are the three sort of ingredients that are critical in an energy transduction process. So exactly how the quinone was interacting with fumarate reductase was important to understand, sort of how electrons that are generated in metabolism are ultimately transferred to fumarate through this complex. But there was also a question, as I recall at the time, whether there could be some steps involved in energy conservation, as respiratory complexes are often involved in. And so the exact location of the binding sites for quinones was important, whether or not this process could be involved in setting up transmembrane proton gradients.
This is probably bigger for me than for Doug. So I was a graduate student at the time, as I mentioned, and it really got me interested in both membrane proteins and how energy is transduced. And this converted, I've kept with understanding how membrane proteins work I'm now a faculty member, and have switched a little bit into how signals are transduced and how information is transduced, sort of stemming from this. But there's also a little bit more. As I read this paper this morning, one thing that I recall is that as a graduate student, I was probably not on the top half of natural writing ability. And I really struggled with my first manuscripts. And this was the first paper, when I reread it, I remember this was the first paper where I really put it together by myself, beginning to end, and I held on to it until I really thought it was right, so that when I finally had a draft that I gave to Doug, he was like, "Oh, this is pretty good." Then he made a couple grammatical errors, but he didn't really have to edit it. And so I remember being really proud of the fact that I'd finally written a paper that wasn't terrible. I don't know of a better word for terrible, in the first few drafts. And it was actually, I was starting to find my voice as a writer. We were talking a little bit before you started the interview that parts of what my lab do now are looking at cofactor insertion, and we continue to use this protein as a model system. We've been recently looking at how the FAD is incorporated into proteins, and FAD is one of the cofactors in this protein and how it gets covalently attached. We are starting to look at how the iron sulfur clusters get inserted and how the whole thing gets assembled, because this protein does have enough complexity that it can provide a model system for a number of things. How do you get a heme inserted into the membrane and associated with the protein? So that was a little bit long-winded, but I think that this work did have a pretty big impact for me.
Yeah, well, I guess in reflecting on this video, I was struck by sort of my career in some odd way has been intertwined with quinones even before I realized it. So in college, I would work summers as a technician at the University of Kentucky in the biochemistry department, and I worked for Professor Robert Lester, who actually as a postdoc had been involved with the first discovery of coenzyme Q when he was at the Enzyme Institute at the University of Washington. And so, in fact, my first paper was with Bob Lester, and it was published in JBC. Then fast forward a few decades, and when we started working on membrane protein structures, our first entry into that was a collaboration with George Fayer's group on the photosynthetic reaction center, which couples the absorption of light to the charge separation and ultimately the reduction of quinones. And so that whole process by which the quinones get reduced and protonated and then leave the membrane was a fascinating project, both scientifically and physiologically, in terms of sort of the role of quinones in the energy metabolism. And certainly fumarate reductase continued that theme, and it turns out just by happenstance, just recently through determining the structure inadvertently of a contaminant on an EM grid, we solved the structure of the ubiquinol oxidase, or we resolved the structure of the ubiquinol oxidase from E. coli, and then trying to find things to look at. We were soaking in quinone analogs, trying to identify the binding site there, since some of the same issues that came up with fumarate reductase appear to be valid for this case as well. So another part of the fumarate reductase project that I have very fond memories of was watching Tina grow as a scientist. She came to graduate school, as I recall, I'm not sure if she knew what she wanted to do, but it certainly wasn't structural biology. And so she came into my group, I think as a late rotation or an additional rotation, and just really had an affinity for membrane protein structure. And it was just really wonderful to see how she developed as a scientist during this time. I don't quite remember the writing experience the same way as I think Tina was always a good writer. And so, but really it was one of the most gratifying parts of being a faculty member is watching when a student comes in and really takes ownership of a project like Tina did on this one, and also several other projects that we haven't discussed, and really finds their voice. I think it's the best part about being a faculty member. So
I can't believe it's 50 years of the PDB. I was actually reflecting on this recently in another context, and realized how forward-thinking it was to have the PDB as compared to many other fields where people put their data in a paper and sort of massage it. The PDB allowed coordinates to be freely available to everyone and basically allowed scientific sharing well ahead of its time in many other fields. And this was when I was a graduate student. I think they were at the 1-year hold phase, and are they going to get rid of the 1-year hold, and how is that going to affect people if you can't sort of claim a little bit of advance sort of working priority on something by giving your coordinates out to someone else. And then not too long later, you had to deposit your data, and now it's becoming more and more common that there's a repository for even the raw data. And as you look at other types of work, they're just starting to catch up, where maybe the supplement of the paper will have like full pictures of the gel, but there's not been the same push to have all data freely available. And I think the PDB was really at the forefront of setting this tone for the community that we are sharing all of our data once we publish it.
Yeah, the PDB was really sort of an inspired development, as Tina was mentioning, and it's now just an indispensable resource. So I can say I started as a graduate student a few years after the PDB began. At that time, to deposit coordinates, you'd actually send magnetic tapes, mail them. And the whole process of even getting IDs, PDB IDs, could take a year or more. And so I think during the period of Tina's paper, that process was speeding up from those early phases, but there were still some of the, I'd say, the charm of the early days. And that was one example of that was that you could, I think, still get custom PDB IDs. And so the original structure that Tina did of fumarate reductase, the PDB ID is 1 FUM for fumarate. And so you were able to do those things. It is the crush of structure determinations and increased. It was no longer possible to be able to get these sort of custom PDB IDs, but it really has changed how structural biologists approach their work, and it's been an invaluable resource for the biological community broadly. And they've managed to keep up with the deluge of structures coming out. So yeah, you can't live without it.
I'd forgotten about the custom IDs. That's been a long time.
Yeah, I mean, so I worked on carboxypeptidase as a graduate student, and at that time, we would have, I guess I can't remember what the original one was. That wasn't my project, but we would have 4 CPA, 5 CPA, 6 CPA for different, different liganded forms and so on. And so Yes, that was sort of nice, but um, you know, pretty quickly and I think that was, people weren't so happy when that phase ended, but in hindsight, it makes perfect sense.